this IB revision video on gene expression and particularly transcription factors, and this is for HL students only. And just remember, this video, certainly in this case, is going to not have enough detail for you to learn everything. You need to look back at each of these areas and make sure you thoroughly understand them for exams. Then what we're going to be looking at. So we're going to be focusing on something called gene expression, which effectively is the turning off or turning on of genes. So it's the process of turning the genotype into the phenotype. So remember, your genotype is what's in your DNA and your phenotype is what proteins are expressed. It's not just generally an on and off process. It can be assessed by measuring quantities of proteins or other gene products. And this links to something called a transcription factor. All cells in eukaryotic organisms have the same genome, but use it differently to function differently. So every cell in my body will have 46 chromosomes with all the genes on there, but not every gene is activated in every cell. And this is the basis of cell differentiation. So in neurons, the genes expressed for proteins will be different from the ones in liver cells. So for example, your liver cells do not make insulin, which your pancreatic cells do. All cells have the same basic housekeeping genes to keep them alive, and they will be activated all the time. And what we're going to be looking at today is something called transcription factors, which decide which genes are being activated. So this is called regulation of protein. This activation is from regulation through proteins binding to DNA sequences. So gene expression requires selective transcription of RNA. RNA polymerase therefore binds to promoter regions of DNA, often in an area called the tartar box because of the letters you can see there. So when we're doing protein synthesis, the RNA polymerase has to bind to the promoter region of DNA in order to start transcription. And a general thing, a protein called a transcription factor attaches to the TATA box to show RNA polymerase where to bind. All genes have similar promoter regions, but only some are used for transcription. And selectivity is dependent on the DNA binding protein. So depending on whether that protein binds or not will depend on whether transcription happens. And how does it attach? Well, DNA double helix has two grooves where a protein can bind. There's a wider major groove and a narrow minor groove, and it normally attaches to the major groove. So some key things you need to understand are that transcription factors are proteins which allow RNA polymerase to bind and thus allow transcription of a gene in protein synthesis. These transcription factors are always upstream of the promoter site. So if the promoter site is here, it will attach up here. Enhancers are then sequences which activate to bind to and increase transcription, and silencers sequences which repressors bind to and decrease the rate of subscription, affecting protein synthesis. So here you can see how the different proteins might work at attach. So you've got the binding sites. We said here the gene is further down here. Okay, we have binding sites, all these tartar sites which the proteins can attach to. We have activator proteins which will trigger DNA transcription, translation, and we have repressor proteins. And you need both activators to be present. So this one would do transcription, this one would have very little transcription, and this one would have no transcription because of the repressor protein. Promoter regions and promoters and repressors, we can also look at regulating translation through degradation of messenger RNA. So you should be aware that transcription produces messenger RNA, and then translation is when messenger RNA is used to put the amino acids in order and at the ribosomes and make proteins, polypeptide chains. So this then is looking at, well, how can the messenger RNA actually be slowly broken down to prevent too much protein being produced? So effectively, you need to know that the messenger RNA from transcription has a long tail of adenine added to the 3' end, and this is called the poly A tail. So that's this bit here. So at the end of the 3' end, with the 5' end over there of the messenger RNA sequence, there is this constant tail of adenine, which you can see here, so one base. This makes it much more stable as a molecule, but this is slowly shortened over time. So what happens is if you have a shorter polypeptide, a poly A tail, your messenger RNA is less likely to be translated over time, and there's an increase in the likelihood it will be degraded by a nuclease enzyme. This basically gets broken down over time, so bits get broken off over time, and this happens over a varying length of time depending on where the molecule is, chemicals present such as hormones. So the shortening rate varies with polypeptides. It can go from anything from 30 nucleotides a minute to 2 per hour. So if we're saying 2 per hour, what we're literally saying is that one is lost, that one is lost every hour, next hour that one is lost, that one is lost, next hour that one's lost, that one's lost. And obviously that one wouldn't last very long. It'll also be something where you need to make the protein but only for a small amount of time. This one would be one that would last a lot longer. So messenger RNA molecules as a result can last between 5 minutes and a week in a cell, depending on what is. Now, in terms of this, obviously the length of persistence can regulate how much protein product a messenger RNA chain can make. Hormones can influence, as I said a few moments ago, and they can make them degrade faster or slower. And an example here is something called vitellogenin, there we go, which is a precursor of egg yolk. So the messenger RNA that makes this protein will last 3.5 times longer if estrogen levels are high than when low, and therefore it's all programmed to obviously last longer, produce more egg yolk at certain times of the menstrual cycle. We're going to be looking at is epigenetics, and this is looking at the influence of the environment or environmental factors such as chemicals on the expression of genes. So to introduce it, multicellular organisms have specialized cells derived from undifferentiated cells. This can happen due to the activation of certain genes and the silencing of others. Epigenetic tags can activate or deactivate genes in DNA, and the DNA base sequence is not changed, so this influences the phenotype, which gene is being expressed, but it doesn't change the genotype. There's some terms we need to know: transcriptome and proteome. So transcriptome is the entire set of messenger RNA that is transcribed in a cell. No cell transcribes all its messenger RNA at once, so the transcriptome is less than RNA copy in each exon. It varies over the lifetime of a cell. And the proteome is the entire set of proteins a cell can produce, not necessarily is producing at that one time. To understand what we're about to talk about, we need to go back to something you should have done during the DNA and biomolecules section of this course, and this is look at the structure of a nucleosome. So if we look at the nucleosome and if we talk about it first of all, and then we'll apply it to epigenetics. So you need to know that the core of a nucleosome contains 8 proteins. So these are proteins here. So you've got 1, 2, 3, 4, 5, 6, 7, 8. This has, there are 2 types of these, 2 of each 4 types, I should say, and the DNA is wrapped around these proteins twice. The DNA goes around once, twice around these 8 proteins in the core. H1 is an additional protein, so this green bit here is an additional protein which reinforces the binding of the DNA in the nucleosome, and it might also help pack proteins when preparing for division and mitosis. And there is a short section of linker DNA binding adjacent nucleosome. So that will attach to the next nucleosome, as will that one. And this is what makes up the structure of a chromosome, effectively. So histone proteins and DNA embedded. Now, why this is important, and I think it's worth showing you here, you can see the DNA gets, when we break it down, to get smaller, and there you go, you can see the histone proteins there in the nucleosomes. Okay, so why this is important is because DNA tags can attach to the DNA and the histone protein, altering how tightly the DNA is wrapped around the histone protein. And this is important because if the DNA is wrapped tightly around the histone protein, the gene is effectively turned off because it cannot be read, and that's called epigenetic silencing. So if you think about it from a vague point of view, if the RNA polymerase can't attach to it, if the bases aren't exposed, that DNA cannot be read. So if it's tightly wrapped around the histone protein, it can't be read. If the DNA is wrapped loosely around the histone protein, the gene is turned on, and the DNA bases then allow transcription. So effectively what we're talking about is what things can affect the how tightly this is wrapped around. And if it's wrapped around tightly, it's turned off. If it's wrapped around loosely, it's turned on. Tags are not fixed like DNA, so it can change due to the environment, resulting in DNA wrapping changes. And this is all about what we call the epigenome, which is the sum of chemical changes to the histone in the DNA, not including base substitutions and mutations of. And this happens during your lifetime. So in early development, signals come from inside the fetus cells and the mother's nutrients to alter the how tightly the DNA is wrapped in, and later on hormones affect it within your life. And environmental factors such as chemicals you've been exposed to, such as if you smoke or if you drink heavily, and UV and lots of things like that can alter this, so the message can signal proteins to move in the nucleus and the genes can become inhibited or activated. Hence, twins start to look different as they get older. So one of the main things that controls how tightly this DNA is wrapped around the histone proteins is called methyl groups. And when methyl groups are added to the DNA, they normally attach to the cytosine in the promoter region of the DNA. And this can cause the genes to be switched off as a result. So that what they do is they suppress transcription, turning the gene off. How do they do this? Well, they prevent the binding of transcription factors to DNA, and they attract proteins which condense the DNA histone complex. If we see here, we've got methyl groups or methylation here, and we're adding it to the histone. So remember, the histone plays a role in the tight packaging of the DNA. So if we add methyl groups, they can attach to the histone tails, making them tighter bonded and meaning less gene expression as a result, because we said earlier the RNA polymerase cannot attach and thus decreases the transcription of the gene. So if we remove the methyl groups, it stimulates transcription, and if we add the methyl groups, it will inhibit. And here you can see the impact by having increase in the methylation, we get more condensed packaging, more tightly packed, and as a result, there's no access to the transcription packed proteins, and the gene becomes inactive. But when we decrease methylation, there is less condensedness of the packaging, and it's more accessible for the DNA polymerase, the RNA polymerase, and then this can activate the gene as a result. So here you can see with the methyl group, they're all very tightly packed and you can't access the acetyl group. Here you can. The area we're going to look at is gene expression and the impact of the environment on that. So continue what we talked about last time. We've looked at methylation. Now we're going to look at some actual specific examples from the environment. To understand different types of twins for this to occur. So there are two different types of twins. There are twins that have developed from the same two sets of eggs and sperm, and they might be the same sex or one of each sex. So here you can see where the egg and the sperm are separate, and therefore they're not what we call non-identical twins. So different egg, different sperm, different egg, different sperm. Identical twins are when one egg and one sperm has fertilized one egg, but then that egg has split into two, leading to identical twins. What needs to happen then is that the epigenome needs to be wiped or reprogrammed for when a zygote is formed. And 99% of the epigenome is erased before making the zygote from the gametes. And this is important because these gamete cells have to be stem cells again. And obviously during your lifetime, the epigenome has made sure that your cells are specialized. Also, any influences of chemicals on you hopefully then won't be passed off to the offspring. Some tags are not removed, however, and this can be passed from parent to offspring, and this is called transgenerational epigenic inheritance. So let's have a look at an example of normally in a recessive disease. If we look at genetic disorders, just with a basic monohybrid cross, you can get the idea that one in every, if you've got two carriers, so my pen isn't working, there we go. This person here is a carrier because they've got a dominant, recessive, dominant, recessive. So if you had a recessive allele, a recessive disease, and you were a carrier, one in every four, there's 1 in 4 chance every child you have would be a sufferer of the disease, and 2 would be carriers, and 1 in 4 chance that you would have a normal child. However, this doesn't. Sometimes there might be an altering in gene expression of the recessive disorders. So normally you'd expect 1 in 4, but in a disease called Angelman syndrome, the dominant working allele may be silenced by an epigenetic tag, and the silence meaning the single copy of the recessive allele could cause the child to have the disease. What we're saying is this person would obviously have the disease anyway, but this one, the dominant has been silenced, and therefore the recessive allele is shown in the phenotype. One of the new studies in the environmental tags is actually air pollution, and we now feel that air pollution is believed to be a major tag in humans. So this could be a key part of the epigenome, which, if you remember the term, is the sum of chemical changes to histone and DNA of the organism. It causes direct damage to tissues, but it's thought to have an impact on protein transcription, resulting in immune deficiency, heart conditions, and lung disease. And large impacts on embryo development in pregnancy because it causes changes to methylation of genes controlling gestation period, which can then lead to premature births and early life development changes. So therefore quite a significant impact that it could have on the development of someone in their whole life. When we study impact of gene expression, the best way to do it is looking at monozygotic twins, identical twins, who start life with identical DNA, and then we can look at how their DNA has changed due to the environmental impact that it's had. The environmental factors can be observed directly. Now, one area where the sort of studies on environmental impact have started is something called the lac operon, which is looking at the production of lactase, which is a key enzyme in bacteria. So this is like an early model that has been used to understand how epigenetics works and how the epigenome works. So if we have a look at this, first of all, explain the different parts of the structure. So this shows you the DNA of bacteria, and we have the Z and Y area of the bacteria here. These are the structural genes. They are the bits that code for the beta-galactosidase and for lactose permease, two enzymes essential in digesting lactose and key in that role. We then have the operator region, which is length of DNA, which can switch them on or off. The promoter region, which is the length of DNA where RNA polymerase binds to transcribe these genes. So it has to bind to this bit here. And I, which is actually much further away, encodes for what we call the repressor protein. So if we were to put the bacteria in a growth medium made of glucose, not lactose, it no longer needs the enzyme that we are just talking about. So we need to be able to stop the production of this because it would waste the energy of the bacteria. So the regulatory gene is expressed, synthesizing something called a repressor protein. So that is obviously happening here, making a repressor protein. That repressor protein binds to the operator region, and that prevents RNA polymerase attaching to the promoter region. This is therefore unable to bind, and therefore RNA polymerase cannot transcribe the genes, and the genes then don't synthesize beta-galactosidase and lactose permease, so the lactose enzymes therefore are not made, which would be a waste of energy. Lactose is present. Lactose itself acts as an inducer, and it binds to the repressor protein, sites on the repressor protein, changing its shape, meaning it will leave the promoter region unblocked. RNA polymerase can then bind to the promoter region, begin transcription of messenger RNA for the Y and Z genes, and this effectively acts as a molecular switch controlling the X and Y in the LAC enzymes. E. coli then can make the two enzymes, lactose permease, which allows the lactose to be uptaken into the cell, and beta-galactosidase, which breaks lactose into glucose and galactose, allowing its use in respiration. So what this shows you is the lactose itself is an environmental inducer that changes the shape. The lactose comes in, changes the shape of the repressor protein, and the repressor protein can no longer attach to the operator region, meaning the RNA polymerase can attach to the promoter region, and Z and Y. And that is really it. So the regulatory gene makes the repressor protein, but when we introduce that environmental impact of the lactose, it will no longer be able to attach, and therefore the enzymes will be able to be transcribed and the gene is expressed.